GCSE Biology Topic by Topic. What we're going to do today is we're going to have a look at photosynthesis. Photosynthesis is probably a key process in the world, because without this nothing sort of survives. We're going to have a look at a couple of experiments which are important for sort of looking at how life really works. So let's get into have a look at what is photosynthesis. Simply, photosynthesis is where plants take carbon dioxide from the air, and with that they take some water, and with this they make glucose, which is C6H12O6, and as a byproduct they get rid of oxygen. If we balance this equation, then 6 molecules of this and 6 molecules of that react to make this, and 6 molecules of oxygen. It's an easy one to remember, 666. And the 6's are also on there, and the only odd one is the H12. So this stuff here is glucose. So what we need to do is have a look at how this works, and we can do this by having a look at a sort of a basic experiment to do this, and we can base all our experiments on this. So let's go and have a look at a simple experimental setup and see what's needed and what's done. So this is the experiment that we look at. I've got a bright light shining on a plant. The plant is set up in a beaker. This is an aquatic plant, and this is out of my pond, and it's photosynthesizing quite nicely, and I can tell that by the fact that it's got lots of bubbles being produced, and it's been going for the last half hour. And if we try to scan up, you can see that in half an hour it's collected a reasonable amount of oxygen, and there, if you saw, was a bubble coming up. We could measure this using a test tube like this, and if I twist it a little bit, you can see on it it's got numbers. Now usually it's supposed to be read that way up, so the numbers are the right way up, but in fact we can turn this upside down and put this over the top of that, and then we could measure in centimeters or millimeters how much oxygen we collected in a certain time. So we can use something like that, but it's usually not accurate enough because it takes basically too long to do it. So what we can do is we can have a look at the bubbles being produced. Now, under an artificial light, and that's what I've got here. I'm just going to turn the artificial light off, and then we can see perhaps a little bit easier. So I've got, in fact, one of my photography lights here, and it puts out a fair amount of light, and I can concentrate that down onto this plant. So our setup is really quite easy. We've just got a plant which we can set up at the bottom, and we put over that a funnel, and over that I put a test tube. Now I've done that all in water, so I needed to put this in to a sink, and filled it right up with water so that I could set it all up. Then I could pour some of the water away and dry it so we could actually use it. So fairly easy to set up, and what we can do is we can have a look at what's actually going on here. I can turn the light back on and the camera just about adjusts, and we've got quite a lot of bubbles going on in here as it's sort of producing a little bit of oxygen. Let's have a look at how I can see that going. So earlier I took a little bit of video of this, and we can see we've got some bubbles being produced. You can see here we're getting bubbles produced. They don't always go straight up. You can see this one's catching and producing a bigger bubble here, and eventually that bigger bubble is going to go up, but it doesn't always go up straight away. So sometimes we're better at trying to identify and just count the number of bubbles that are sort of coming from here. We then have very little idea about the size, but they seem, looking at them, to be fairly consistent. And being fairly consistent, we can sort of use that as a way of trying to do some of the measurements. So it's sort of not a bad way, I suppose, of trying to measure photosynthesis, and this then is often used as a practical to sort of measure how plants work, and deals largely with aquatic plants, but it does need a fair amount of light to try and get these photosynthesizing really well. And then there are some problems, of course, that will do that. So let's have a look and see where it all starts. And I've got here a photomicrograph of a piece of grass that I cut, and I've cut it across the piece of grass, just so that you understand what I mean by that. I took a blade of grass and I've cut it across there twice, once there and then again another one just underneath that one, so it's a very thin slice, and I've mounted that onto a slide. And we can see various interesting things about this blade of grass. It's got some cells on the outside which look clear. Underneath that we've got some cells which are very green. I've got some cells here and there. These are vascular bundles. This is where the xylem and phloem are that transport the water, and then inside I've got some storage space where this leaf will store various bits and pieces that it's made. Not the glucose, but it tends to store this as starch. If I look in closer, then we can see that some of these cells are absolutely filled with these little green blobs. These are the chloroplasts, and perhaps you can see a little bit clearer in this one some of the chloroplasts inside one of these cells. And that's about the maximum magnification my microscope can go to produce these sorts of images. What I can do is I can look at a whole variety of different bits and pieces. I'm just going to take you to have a look at another one, and we're going to have a quick look at this. This is the outside of a pine leaf just off of a Christmas tree, and here we can see rows of holes in this leaf. These rows of holes are how the oxygen gets out and how carbon dioxide gets in. These are the stomata. This is a cut across a leaf of a piece of pineapple, and here we can see a vascular bundle. These are the phloem cells, these are the xylem cells. So the xylem cells bringing water to the plant, and the phloem cells taking the glucose away. Not much in the way of greenery here, but if we go out, then we can see more vascular bundles, and there I've got some cells containing a lot less chloroplasts in them, but nevertheless you can see individual chloroplasts inside, in this case, a pineapple leaf. So what we want to do is to have a look at a variety of experiments that we can use to work out what's going on in plants, and let's explore this in more detail now. The first thing I suppose we need to do is have a look at the structure of a leaf, so we've got a better idea. And a leaf will have typically, this is a very general leaf, some cells at the top. These tend to be clear, and these cells make the upper layer of a leaf. And these are the epithelial cells. Now on some plants we have above this a waxy cuticle, a watertight layer. Underneath this we have a row of fairly long thin cells, and these make the palisade cells, and it's these that do basically the main photosynthesis, and these are jam-packed with these chloroplasts. Underneath this we have a layer of cells, and we might have a vascular bundle. This is going to have a little layer of cells going round, small cells, which are the phloem, and much larger cells which make the xylem. And then we continue with these cells across, and deliberately you can see there are spaces here. The spaces here simply are part of this spongy mesophyll. The spongy mesophyll cells allow air to move around. Water comes up through the xylem, leaks out here, and water moves around. Water required for the photosynthesis, and we want to release the oxygen produced and take in carbon dioxide. So we have a lower set of epithelial cells, and with these we often have associated with these little holes. The hole is a stoma, and many of them equal the stomata. The special cells either side are called guard cells, and these guard cells can open and close to let in or let out more water and carbon dioxide. So this is our basic structure of a very typical leaf. So we can look at various properties of these leaves. The properties of our leaves, typically they are broad. This gives them a large surface area for photosynthesis, where an enormous amount of light can fall on. Typically, most leaves are also thin. They're thin so that the gas can diffuse across them, and this is quite a thin layer. I've expanded this quite a lot to show you what's in there. Well, these cells also contain the chemical chlorophyll, and this chlorophyll is contained in little containers called chloroplasts. And the chloroplasts, yeah, these are those green little containers. So the green container, a chloroplast containing the chemical chlorophyll, which is used to split water into oxygen and hydrogen so that it can be combined with the carbon dioxide to make glucose. We've got in these cells veins, sometimes called a vascular bundle, and this is made up from the xylem cells that bring in the water and the phloem cells that take away the produced sugar and sends it round the rest of the plant. We've got large air spaces. The large air spaces in a plant are to allow carbon dioxide to get into the cells and oxygen to leave, and all that's done by this process of diffusion. Then we've also got typically the stoma with its guard cells around it, and these guard cells open and close the stomata basically to regulate the amount of gas that can get in and out and the amount of water that can leave. So all of these adaptations are done and designed to allow the plant to get the maximum amount of light. Certain algae are aquatic. They're capable therefore of photosynthesizing in water, and like the plant that I've got there, which is a sort of pondweed, so that it can sort of survive and grow quite nicely in sort of simple aquatic conditions. So we can know a few other things, and from chemistry we can work out that photosynthesis is an endothermic reaction. It takes in energy, and in this case it takes in energy in the form of light from the sun. During photosynthesis, that energy is transferred from the environment to the chloroplast, and the chloroplast here uses that energy to convert carbon dioxide and water into sugar, and we get oxygen produced as a byproduct. So the oxygen really not wanted by the plant, and over the millennia where plants have lived on the earth, especially before the time of animals really living much on the earth, they converted an awful lot of the carbon dioxide in the air into oxygen, although most of that was probably done by the photosynthetic bacteria that came before them. So there's our idea of the process of photosynthesis. It's really quite simple, and let's have a look then in more detail about what we can do to try and work out what happens. So our first job would be to look at something about here the rate of photosynthesis. Now, in my experiment, I am normally quite grateful that I can actually get this to work at all. And we need vast amounts of light going to try and get this working properly. But what we can do with this is we can do an experiment to see how this light affects this plant. And what I can do is, by using something like a simple here as a ruler, I can set this to be, in this case, 30 centimeters away from the plant, and I can try this to see how it is if I move it further away. And what we would have to do with an experiment like this is I would have to set this up and leave it for a considerable amount of time. So I'd set this up, leave it for, let's say, an hour, and I can collect the amount of oxygen. Then I could perhaps repeat this experiment, but at a further distance away. Now, as I move it a further distance away from the light, less light comes onto the plant, and you can see this as it's set up here. If I look at this bit here, there's a lot of light on my hand. But as I move my hand further away, you can see there's a pool of light here, and it's gradually getting less and less and less. So by the time I'm out here, we're getting an awful lot less light falling on these leaves. And because of that, it's not going to photosynthesize as well, and therefore it's going to produce a lot less oxygen. When I normally set this experiment up, I like to have the plants photosynthesizing quite well. So what we normally do is we set this up and we leave them out in the sunshine for a while, which is what this one was doing. And as you saw in the video, as it was going, it was producing quite a lot of bubbles, whereas if I look at this now, it's going to produce a lot less. And if I get down and have a look at this little thing producing a bubble, whereas outside you saw it bubbling a lot, here if I just watch this produce a bubble, well, in all that time it didn't manage to produce a bubble at all. So it is going, and you can see a lot of these little bubbles just basically stay attached to the leaf, and it's always a little bit of a problem, this experiment. We can see the tiny bubbles rise to the surface, but it's not particularly easy to get some of them to come up. I'm just dislodging bubbles that have been made over a period of time. And so this is where it's not really accurate trying to do this, and we need to try a better system, and that is usually trying to count those tiny little bubbles being made from a particular piece of the plant. So the obvious thing that affects photosynthesis is light. And grabbing his ruler, we've got a simple system that we can set up. So I've got my distance here. I'm going to set up my light, and this light we can put at a particular distance away from my plant. There we are. Let's just draw in the funnel and my test tube and my layer of water there. So with the plant sitting in here, we can set up and measure and record basically the distance here, and we can try to work out something about the light intensity and the rate of photosynthesis. And as I've said, the rate of photosynthesis is easier to record if we use and measure simply the bubbles coming off. And going back to the video there, we can see and measure here the bubbles coming off, and simply by putting a stopwatch there we can actually measure how many bubbles coming off at a certain time. And measuring the number of bubbles coming out is probably a more accurate way of trying to do this. If we leave it over a considerable amount of time, then we'll also see that what we've got, we can collect the amount of oxygen, we can collect that in that sort of test tube I showed you before. Which has these sort of measurements, and you'll see that because of the shape of this test tube we can actually measure small amounts relatively accurately. And I didn't use this one in this experiment simply because I wanted you to see a normal test tube, which is what you normally get. So if we do it for a very long time, in the means of hours here, then we can collect and measure the amount of oxygen, and it is largely oxygen in there. If we're going to do it for a shorter period of time, then we count the bubbles. So over a large time period, then I would simply collect the O2 with basically some sort of measuring. Here I use a test tube. If I'm doing it over a short period of time, then I would count the bubbles. But it's got to be from the same point. So how does light affect? Well, let's try and see if we can draw this out and show you. What I've got is my little light source, and it's shining out here, and let's set this to be 1. Now this is my single distance here, whatever it's going to be. This is my distance, and we get a certain amount of light falling on the plant. As we move this further away, and we have to try and think of this basically then in sort of 3D, what I've got is that by the time we double this distance, and that's about 3 centimeters, so let's go to about 6 centimeters here. By the time I get to this distance here, as we basically double the distance, then instead of having one area, I could probably see that this is now about 4 times the area. And this is called the inverse square law. The inverse square law is that basically we can say that the light intensity is proportional to 1 over the distance. Now, what does this mean? So this means if you double the distance between the light and the plant, here, going from there to there, then the light intensity falls by 1/4, because this distance here is squared. So what this means is that as we increase the distance just a little bit, the amount of light falling on this decreases by an enormous amount. So this is all about light intensity and the inverse square law. We don't have a linear relationship as you move it, so it becomes less. It's in fact decreasing with the square of the distance. So moving a small distance can have a massive effect on the amount of light falling on the system. Now, what we will see if we do this experiment, and it's not easy doing this with the time available, we can see that as I move this, then the amount of photosynthesis decreases. And let's see the sort of graph that I would get with this. And basically, as I move this away, so this is going to be my rate of photosynthesis, and here I'm measuring my light intensity. Now, measuring the light intensity can be quite difficult, and you wonder, well, basically, how can I go about measuring the light intensity? It's going to require something quite complicated. No, it isn't. All we need is something like a phone, and what we can do is we can use a phone to measure the light intensity. And there are a whole series of different apps that we can get on these little phones, so that you can manage to get a measure of the light intensity. And typically the one that I wanted to show you has disappeared. There we are, I've got one. And what I can do with one of these is I can get some sort of measurement of the light intensity. And this is a photographic one, and it measures this in camera settings, and that's what I use. That one's called Lux. But there are other ways that we can do this, and there are other little meters that we've got that we can use to sort of measure the light intensity. So photographers would use this little thing to measure the light intensity, and I'm just going to get my son to get the top box out, and he's going to bring me a light intensity meter so I can show you what one of those looks like. No, not there, Paul. By the bookcase, the top of the 3 black boxes. And it comes brilliantly, because basically I forgot to get this one out. And here I've got my little meter, and all I need to do is to plug in the little meter, and what it's got is a little sensor. So if I put this down here and we switch it on, I've got a measurement here in lux of the amount of light falling on this. So at the moment we've got that. If I turn a light on top of this, then you can see it's going to go up. Turn the light off, it's going to go down. So we can measure the amount of light. And if I go and measure that on this plant, then we can measure the amount of lux falling on this. And if I put there, we've got 450 lux falling on it. If I double its distance, so at the moment that is at 15 centimeters. let's double that to 30 centimeters. Then you can see that our lux here, if I put it right in front of the lights, that was 1200. Bring it down here, it's only going to get to about 500. If we now increase the distance again, my scales were changing. So there's my about 1200. Halfway came down to about 500-600, and then the scales drops down by a factor of 10. So this is now 1 point in the same manner, so it's about 1 point something. So my lux meter will measure what's going on quite happily with the plant, and it can be used to get a much better reading of what's going on. But you've got to be careful with these things about things like scales. When I take this outside and measure, I get vast differences, and you think that a room is actually quite bright, and here I'm looking at about 250 lux here, sort of varying quite a bit. So there we are, 320 lux there, whereas if I take this outside, we find we might go up sort of 2, 3, 4, 5,000. Gives us quite a difference with the amount of light. You don't seem to appreciate, your eyes sort of adjust, and so you don't see the difference in the light, whereas a meter gives a much better impression of what it is that it's doing. Just put my light meter away. Or not. So we're seeing that the light makes a change here, but the rate falls off dramatically. That here we've got light sort of being really an important factor, but as soon as we get down here, we're losing a terrific amount of light. So light is very much a limiting factor. The brighter the light, the faster the rate of photosynthesis. What other factors might affect light, affect photosynthesis, as well as light? Well, there are two other factors that are really quite important. So the first of these is temperature. We know that when it's cold, plants don't photosynthesize very much, simply because this is all controlled by enzymes, and plants work probably best at probably around about 20 degrees centigrade Celsius. They don't particularly like, really, in this country, a lot hotter temperatures than that. They work about that the most efficiently, and they're all right up to around about 40 Celsius, but above that then we get the enzymes being denatured. So we're looking at that sort of temperature. But if we try to see what that looks like with a temperature curve, then it's going to increase and increase and increase, and then we'd have a sudden fall off. And this sort of fall off point is about 40 Celsius. Below zero, we don't tend to get any photosynthesis taking place because the enzymes then don't work if everything's in solid. The other thing that can make a difference, and that's something I haven't taken into account with my experiment, is the amount of carbon dioxide. Now, in my experiment that I've done, I've simply taken my plant and I've left my plant, camera's in the wrong place, in light, but it's just in water. Now, if I want to and worry about this, then we need to actually increase the amount of CO2 in here. And how do I increase the amount of CO2 in there? Well, it's very simple. We have to add CO2. And in my fish tank, I would in fact bubble in some CO2 and I leave it to sort of come in to the plants. I literally have an extra cylinder of CO2, and this makes the world of difference to the plants. They grow much more vigorously, and as you know, if you've got an aquarium at home, then you normally have some lights in there, and the lights, yeah, they are for the fish, but mainly they're there for the plants. The fish, yeah, they like to see, but basically you want a lot of light in there to help the plants photosynthesize. To get more carbon dioxide, then what we need to do is put perhaps some chemicals into the system. Normally carbon dioxide 0.04%, and what I can do is I can increase this by perhaps adding some chemicals that release carbon dioxide, and that gives me a graph again very similar to the light intensity, which then we've got CO2 concentration, and again I've got my rate of photosynthesis. So as I increase the carbon dioxide, then the rate of photosynthesis will increase, but after a while increasing it more doesn't make much difference. Why? Because it's also being affected by things like light, temperature as well. So there are a whole series of experiments that you can come up with by putting in different amounts of bicarbonate solution that will release carbon dioxide. You can try different concentrations of that, you can try different temperatures, you can try different distances, all on that same setup that we've got of having this set up with a light. So there we are, that's photosynthesis, an idea of trying to do things and measure the rate of photosynthesis. If you like this, then please subscribe and give us the thumbs up, and I'll see you next time when we'll do some more work on how plants use the products of photosynthesis to grow. So I'll see you next time on GCSE Biology Topic by Topic. Until then, stay safe. Bye bye.